A single protein can reverse the developmental clock on adult brain cells called astrocytes, morphing them into stem-like cells that produce neurons and other cell types, UT Southwestern researchers report in a PNAS study. The findings might someday lead to a way to regenerate brain tissue after disease or injury. We're showing that it may be possible to reprogram the fate of this subset of brain cells, giving them the potential to rebuild the damaged brain, said study leader and co-corresponding author Chun-Li Zhang, Ph.D., professor of molecular biology and an investigator in the Peter O'Donnell Jr. Brain Institute. During development, mammalian stem cells readily proliferate to produce neurons throughout the brain and cells, called Glia, that help support them. Only two small regenerative zones, or niches, remain in the adult brain, Dr. Zhang explained, leaving it with extremely limited capacity to heal itself following injury or disease. Recent research has suggested that Glia can be prompted to produce neurons in some models of brain injury or after genetic manipulation. Although these findings are promising, regenerating healthy brain tissue will require production of multiple cell types, rather than only neurons, said Dr. Zhang. Looking for a way to spur this multipotent regeneration, Dr. Zhang and his colleagues used a genetic engineering technique in adult mouse brains to induce astrocytes, a subset of Glia, to produce different transcription factors, proteins pivotal for controlling cell identity. These experiments showed that a single transcription factor, a protein known as DLX2, appeared to reprogram astrocytes into neural stem-like cells capable of producing neurons and multiple subtypes of glial cells. Working with the team of co-corresponding author Gary Hun, Ph.D., assistant professor of obstetrics and gynecology and in the Cecil H. and Ida Green Center for Reproductive Biology Sciences and the Lyda Hill Department of Bioinformatics, global gene expression analysis showed that prompting a stroke. Researchers in the Zhang lab are planning to study this approach in animal models. Current UT Southwestern researchers who contributed to this study include Sergio Cananzi, Lei Wang, and Yuha Zhou. Other participants include co-lead authors Boxin Li, now at Duke University, Yunjia Zhang, now at the Beijing Genomics Institute, China, Xuanhui Han, now at Peking University, China, and Yongxin Fu, now at Tsinghua University, China. This research was supported by funding from the Welch Foundation, I-1724 and I-1926201700325, the Detchard Foundation, the Texas Alzheimer's Research and Care Consortium, TARCC 2020, the Kent Waldrop Foundation Center for Basic Research on Nerve Growth and Regeneration, the National Institutes of He. https://ift. TT, S5Z7 al., accessed March 8, 2022. Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you.